Now that we have a sense of the scale of the human genome, let's talk about how the genome is transmitted from one generation to the next. And this represents the last installment of the lectures where I studiously avoid talking about genetic variation. Paradoxically so, because when you talk about passing something on from one generation to the next, why wouldn't you talk about genetic variation? Well, because I really want to keep the mechanism of the transmission separate from the idea of variation. And so we're going to talk about the mechanism of transmission. I showed you this picture before, the 40 or so cell divisions in a single lineage that lead from one generation to the next. Here is you, and you have to imagine 100 trillion cells that all emanate from these cell divisions. And here was your dad. And of course, your dad contributed a single cell to you that had only one genome. This sperm here didn't have both genomes from that your dad carried, and only had one genome. And so the process by which, there needs to be a process by which the diploid genome is reduced to a haploid genome. It's called meiosis. I won't get into the details of meiosis because the mechanisms are really elaborate, as you might imagine, and very interesting. But the end result is just that one chromosome is passed on, or one set of chromosomes is passed on. And that's the result that matters. So we're going to talk about how we're going from one genome from dad and one genome from mom to the diploid state, and then back to a single genome that gets transmitted. Okay, so this picture should be really quite obvious at this point. We are diploid, one genome from mom, one genome from dad, and that's how that happens. Okay. Now, each chromosome, you get one copy from mom, one copy from dad. And that is really, again, I've said it so many times, it really should be obvious by now. And of course, once he contributes his genome to this, she does the same thing. Now, the way this happens here is that you could pick, in principle, you not just could pick, you do pick, in principle, for what goes in from the dad's side, you pick one of these, okay? So you pick blue here, you might pick blue here, you might pick blue here, okay? So you get blue, 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 okay? In other words, 3 chromosomes that all came from grandfather. But of course, at every time, at every chromosome, you have the choice of picking one or the other. And so if we only had 3 chromosomes, we would have 8 possible configurations that you would have 8 possible sperm, if you will, that would contribute to the next generation. Okay, so in the first generation, it's really straightforward. Pick one from dad, pick one from mom, put them together. It gets interesting in the generation after that, when it's not just about looking at your mom and your dad, but when it's about looking at your grandparents. And of course, that's what happens on this side, and exactly the same thing happens over there on that side of the family. Okay, so same picking occurs, and mom contributes the genome. It gets more complicated because what happens during meiosis, when the germ cells are being generated, is that the chromosomes don't remain distinct as paternal and maternal, but you have a process called recombination. Recombination occurs such that the chromosomes are literally, your mom and dad chromosomes are literally lined up with one another, and there's a crossover that hooks up a portion of dad's chromosome with a portion of mom's chromosome. And then they switch. And so now you get sort of a hybrid chromosome, if you will, where this piece here comes from grandfather with respect to this person here. This piece comes from granddad, and this piece comes from grandmom, right? So recombination further increases the universe of possibilities, if you will, in the second generation. Recombination likelihood or probability or frequency is higher at the chromosome ends. This is a technical detail, but it's worth keeping in mind. And lower sort of towards the middle of the chromosome. And per chromosome, I've drawn here a standard size chromosome that's perhaps 100 million base pairs long. Per chromosome, there is on the order of 1 to 2 recombination events. Across the entire length of the human genome, there are approximately, let me do the math really quick, 3 gigabases, 3,000 centimorgans, 30. There are 30 recombination events distributed across all human chromosomes in each germ cell. And again, partly the point of this is to show you that the probability of recombination is higher towards the end. The other corollary, of course, is that there are not highly specific places on the chromosome where recombination occurs, there are, but they are at the base pair level. But in terms of the distribution, it's not just recombination occurs always here, but rather it can occur along the length of the chromosome. And the specifics of exactly where it occurs is very fine-grained at the base pair level. So there is some preference for particular regions, small regions where recombination occurs, but overall there are many such regions that are candidates, if you will. And the genome basically, the cell biological machinery basically picks amongst them randomly, more or less, given this distribution. So from the perspective of the individual, let me draw it out this way. Let's say we're male, and I apologize for this formatting error here, which will keep occurring. We have a maternal chromosome from mom and a paternal chromosome for each of the chromosomes. And if you remember, we have 22 of those. I'm only drawing out 3 of them. And then, of course, there's the sex chromosome, the X and the Y. So in the case of a male, you have a Y chromosome. Looks the same in a female, except in the case of a female, you have 2 X chromosomes. And this gives me the opportunity to digress here really quick, because sex chromosomes are just fascinating. So let me tell you about the Y chromosome. It turns out that the Y chromosome is structurally different. It really contains a heck of a lot of different DNA sequences from the X chromosome. That structural difference is essentially an evolutionary artifact. It is not the entire Y chromosome that makes us male. It is rather a single transcription factor on the Y chromosome that makes us male. So recall that a transcription factor is a regulatory protein that binds DNA in a sequence-specific manner at a time when it is required. So this factor, called SRY, is turned on early in embryogenesis in all cells. And it turns on a series of a collection of downstream genes that give the male phenotype. That produces, higher levels of male hormones, also represses certain female hormones, and so forth. The enzymes that make those hormones, excuse me. So there's a whole battery of downstream events that happen when this transcription factor is turned on. And that transcription factor happens to live on the Y chromosome. the fact that the Y chromosome is structurally different is a product of evolution, and I could go on and on about that. It's fascinating. But let me just mention that the fact that sex is just a gene regulatory effect plays out in the animal kingdom over and over again. For example, in fish, there are certain fish where the biggest female changes its sex to become the male. Okay, and if that male dies, then the biggest female of the group changes its sex to then become the male. Okay, so male, female, at least in those fish, is basically a gene regulatory thing that can be switched on and off. And in fact, in many, many non-mammalian organisms, there is no difference between the so-called autosomes, the run-of-the-mill chromosomes, and the sex chromosomes. As far as most of the DNA sequence is concerned, the only difference is that SRY gene that flips the switch between male and female. Okay, so we always talk about the Y chromosome as sex determining. That's only true to the extent that the Y chromosome happens to have SRY, that transcription factor that flips the switch on a bunch of genes that then turn on the male pathway. If you don't have SRY, you default to the female pathway. Another brief gene regulatory digression has to do with this difference here, where males have one X chromosome and females have two X chromosomes. When the autosomes are all present in equal numbers between males and females, right? I mean, one from mom and one from dad, two every time. Here we have two X's in females and one X in males. So what might happen is that you have twice as much RNA produced from all of the active genes from the X's, because you have two X chromosomes, than in males. When that happens on a large scale, things go badly awry. So in order for the physiology of the organism to be maintained properly, and for the homeostasis and all the gene regulatory stuff to be happening correctly, you can't have an entire chromosome worth of genes expressed at twice the level as in your brother, okay? So what does the female cell do? It inactivates an entire X chromosome with few minor exceptions. There are genes that escape X inactivation. That's few and far in between. So basically, both males and females have one active X chromosome. Males, because they only have one X. Females, because they actually turn one off completely. So every cell in a female has a heterochromatinized, packaged away X chromosome that it stuffs into the corner, literally, and only one of them is active. Okay, so that was a digression into gene regulation as far as transmission genetics is concerned. Let's get back to recombination real quick. So recall that, let me just go back really quick here. Recall that the grandparental chromosomes can cross over and recombine such that you basically have a hybrid chromosome from grandpa and grandma. And there are approximately 30 such recombination events scattered across the genome in humans in every generation. So from the perspective of the individual, let's extend the labels on the chromosomes to the grandparents. So again, let me remind you, I'm labeling only 3 chromosomes, one of them from mom, one of them from dad. And now we're going to also label from the perspective of this grandchild, the grandparental chromosomes, and we're only going to show the ones that made it into the parent. Okay, so of course, grandpa here also has a chromosome from mom and a chromosome from dad, right? But we're going to ignore that. Otherwise, it would get too complicated. we're only going to show the chromosome that makes it into pop from grandpa and the chromosome that makes it into pop from grandma, and same on the female side. All right, so now we have these labeled with colors. Same, and these colors will appear in all of the chromosomes, okay? So while I'm only drawing one chromosome here, I should really be drawing 3 that are orange, just to keep track of which grandparent comes from. So when you have recombination, this is what you will then get. So maternal chromosome 1, so the chromosome 1 that comes from mom, is a recombination product of the 2 grandparental chromosomes that made it into mom. And the recombination event can be anywhere along the length of the chromosome. And so I've just drawn one arbitrarily here, right there. Okay, 1/3 of the chromosome comes from grandpa on the maternal side. 2/3 of the chromosome come from grandma on the maternal side. And the same concept over here with a paternal chromosome 1, paternal with respect to this person here, so the father. About half of the paternal chromosome 1 came from grandma, that's the green piece, and about half came from grandpa. And this happens separately in every chromosome. And of course, the chromosomes don't talk to one another. The places where these recombination events happen are always randomly drawn from the length of the chromosome. And sometimes there are several, and sometimes there are none. So for example, here I've drawn a chromosome 2 that is from the maternal side. And if you look, this upper portion of chromosome 2 came from grandpa, then there's a portion that came from grandma, and then there's another portion that came from grandpa. And that's because in the germline of mom, on this chromosome, there were 2 recombination events. And then the product of that recombination was passed on to this individual. Okay, so this is just one possibility. This is another possibility. Oh, by the way, here's chromosome 3 where we have 1, 2, 3 recombination events, and I don't have drawn any. Oh, here's one drawn where there's no recombination event by chance. The number of recombination events scales with the size of the chromosome, as you might expect. So chromosome 1 being the largest one generally has the highest number of recombination events. Chromosome 22 often doesn't have any. But it's all independent from chromosome to chromosome and randomly positioned along the length of the chromosome. And finally, here's yet another variation. I mean, there's an infinite number of variations you could come up with. So while you get a chromosome from dad and a chromosome from mom, it gets interesting once you look back one generation further and you really begin to see how the different chromosomes recombine and how you get to certain regions of the genome that come from your grandparents. And of course, if you went back further and further, that would break up further and recombine more and more. So chromosome transmission, summary. For each autosome, you have one copy from dad and one copy from mom. If you're female, you have a copy of the X from mom and a copy of the X from dad. So basically, in the female, the X chromosome behaves like an autosome. If you are male, then of course your Y comes from dad by definition, right? Dad wasn't female, dad was male, and therefore he had a Y. So the Y always comes from dad, which means that your X always comes from mom if you're male, okay? That gives rise to some very interesting genetics later on. So that's the exception. Recombination and independent assortment shuffle your grandparents' contributions in very interesting ways, and it essentially make it so that different portions of the different chromosomes have essentially an infinite number of possibilities of coming together. So, in summary, this set of lectures covered the information machinery and how the genome is organized, and then how the chromosomes are transmitted to the next generation. We have succeeded in not mentioning genetic variation at all. And so next time we're going to, given all of this machinery, next time we're going to talk about how genetic variation arises and how it is transmitted, and about the dynamics of the variation in the population.